Assalamu alaikum dear students. As you know, we are continuing the topic of fermenter. We have discussed the two types of fermenters, batch fermenters used for batch fermentation and continuous fermentation. Next topic is advantages of using fermenters. Very important long question as well. The question may be asked, write the importance of fermenter. The answer would be, a fermenter optimizes the growth of the organisms by controlling many factors like nutrients, oxygen, growth inhibitors, pH and temperature. This is the importance of using fermenter. So the MCQs include, a controlled environment is provided by fermenters. A fermenter may hold several thousand liters of growth medium. Using the fermenters prove much inexpensive is the last MCQ on this page. Next topic is genetic engineering. It is also very important long question, students. Define genetic engineering. Genetic engineering or recombinant DNA technology involves the artificial synthesis, modification, removal, addition and repair of genetic material or DNA. Important MCQs include, the definition of genetic engineering may be selected as an important MCQ. The second name for genetic engineering is recombinant DNA technology. Genetic engineering developed in mid-1970s. It is possible to obtain millions of copies of a specific DNA inside a bacterial cell by genetic engineering. Students, next topic is objectives of genetic engineering. It is very important long question. Then the answer of this question, write the objectives of genetic engineering starting from here and it ends over here. Talking about the MCQs, we discussed the first objective of genetic engineering is isolation of a particular gene or part of gene for various purposes such as gene therapy. Another objective includes production of particular RNA and protein molecules. The next topic is basic steps in genetic engineering. So students, the first step in genetic engineering is isolation of gene of interest. Define restriction endonuclease is the first question. Special enzymes called restriction endonucleases are used to cut the identified gene from the total DNA of donor organism. Second step is insertion of the gene into a vector. Question may be asked in the form of, name the vectors used in genetic engineering. Two vectors have been discussed over here which are used in genetic engineering. First is the plasmid and second is the bacteriophage. This is very important short question. Define plasmid. Plasmid is the extra chromosomal DNA present in many bacteria. Define recombinant DNA. The vector DNA and the attached gene of interest are collectively called recombinant DNA. So MCQ may be asked in the form of, two enzymes are used. The gene of interest is attached with the vector DNA by using endonuclease, that is the breaking enzyme, and ligase, that is the joining, and the second name for ligase is the joining enzyme. The definition of recombinant DNA may be taken, may be selected in the form of, may be selected as an important MCQ too. The next steps include transfer of recombinant DNA into host organism, growth of GMO and expression of the gene. What does GMO stand for? GMO stand for genetically modified organism. The next topic is achievements of genetic engineering. Students, the achievements in genetic engineering may be selected as an important long question in board exams. And if we talk about the MCQs, then the first MCQ is the enzyme urokinase is used to dissolve blood clots. Genetic engineering techniques can also be used to cure blood diseases like thalassemia and sickle cell anemia, which results from defects in single genes. Genetic engineers have developed plants that can fix nitrogen directly from atmosphere. Such plants need less fertilizers. And very important MCQ, in 1980 interferon was produced in the genetically modified microorganism for the first time. Students, it is the diagrammatical representation of production of insulin through genetic engineering. So if we are talking about the achievements in genetic engineering, then insulin may be produced by using genetic engineering. Then you have to draw this diagram. Alright students, now we are moving towards the last question, very important long question as well, single cell protein of this chapter. First of all, define SCP, or what does SCP stand for? SCP stands for sickle cell protein, and it refers to the protein content extracted from pure or mixed cultures of algae, yeast, fungi or bacteria. Next question is how SCP may be produced. For the production of SCP or single cell proteins, the microorganisms are grown in fermenters. These microorganisms utilize a variety of substrates like agriculture waste, industrial waste, natural gas like methane, etc. Microorganisms grow very vigorously and produce a high yield of protein. Define mini-food is the next question. The protein content produced by microorganism is also known as novel protein or mini-food. These were the 3 short questions under discussion. Moving towards the MCQs, the definition of SCP may be selected as an important MCQ as well. Then likewise the definition of mini-food or novel protein. Students, next question is, which scientist introduced the technique of SCP? This technique was introduced by Professor Shrimshaw of Massachusetts Institute of Technology. Write the significance of SCP. Then the significance starts from all scientists recognize. Then there is the significance of SCP. If we talk about the MCQs, then the name of this scientist, Professor Shrimshaw, may be asked in the form of MCQs, who introduced the concept of SCP. Other MCQs include the microorganisms grow very vigorously and produce a high yield. It has been calculated that 50 kg of yeast produces about 250 tons of protein within 24 hours. Algae grown in ponds produce 20 tons of protein per acre per year. This yield of protein is 10 to 15 times higher than soybean and 20 to 50 times higher than corn. These were the important MCQs on this page. Well students, the significance of SCP is also continuing over here. And if we talk about the MCQs, then in the production of single cell protein, industrial waste are used as raw material for microorganisms. It helps in controlling pollution. Other MCQs include production of single cell proteins is independent of seasonal variation. Very important MCQ. It is known as single cell protein because the microorganisms used as producers are unicellular or filamentous individuals. The question may be asked from this block like why single cell proteins are named so. Then the answer would be there. SCP is gaining popularity day by day because it requires limited land area for production. This is the last MCQ. Alright students, we have completed the chapter biotechnology of your biology 10th textbooks. Alhamdulillah. The important short questions and MCQs have been indicated in this chapter. Okay, thank you very much students.